I'm going to start with the very first workshop presentation with the aspiration of explaining single-cell mass spectrometry analysis in a very accessible way, I hope, particularly from the perspective of somebody who is very experienced with single-cell RNA sequencing, with other forms of single-cell analysis, but perhaps not with single-cell mass spectrometry, with the idea being to provide the 10,000 feet overview and the framework within which you can put in context all of the exciting discoveries that you're going to hear about and method developments that you're going to hear about from colleagues presenting throughout the rest of the meeting. And I will focus on mass spectrometry, not because we are not interested in all the other methods, we very much are, but we want to make this field that has a substantial amount of jargon accessible to the broader community. And why do single-cell mass spectrometry instead of using some of the other more mature tools, such as affinity reagents? Some of the reasons for that include the higher specificity that we can achieve with single-cell mass spectrometry for detecting proteins, being confident that the signal really comes from the analyte that we're measuring, and the potential to measure much larger number of proteins per sample. But ultimately, we're not interested just in protein abundances. One of the biggest advantages of mass spectrometry measurements in single cells, in my mind, is the possibility to extend the analysis to the various forms of modifications of proteins, such as post-translational modifications, proteolytic cleavage as used during regulatory signaling in cells, and you're going to see examples for that during this meeting, to be able to measure dynamics of proteins, degradation and synthesis, and all of these other aspects that are really important for biology, because at the end of the day, measuring the abundance of analytes, whether they're RNAs or proteins or metabolites, it's as useful as it is, it's limited. These analytes interact and they become live, they give rise to life because of all of these higher orders of interactions. And mass spectrometry could open the window towards exploring these higher levels of regulation. So I'll start my introduction of single-cell mass spec from the very beginning with sample preparation, which had been a major limitation for a long time. Certainly when we started this meeting, the first meeting, sample preparation was one of the main bottlenecks. What is fortunate to acknowledge now, looking back, is that the community has developed a number of methods to do sample preparation to the point that I think it's no longer the bottleneck. And these methods can be grouped into several categories. One category could be sampling cells with microcapillaries, and this could be patch clamp or could be capillaries of different kinds. Another set of methods use multi-well plates, such as 384 well plates. And these methods, for those of you familiar with single-cell RNA sequencing, are analogous to methods for SMART-seq or cell-seq. They're not identical, there are important differences, but conceptually it is similar idea. You isolate cells via FACS sorting usually and do the preparation into a multi-well plate. And then there are methods that can do the preparation on flat surfaces. And Andrew Leduc is going to talk in many more details about these methods after me. We are particularly excited about the surface droplet approach because we think it can scale the analysis of proteins in the same way that the in-drops or drop-seq scale the analysis of single-cell RNA sequencing. One important point to mention for sample preparation that is different from single-cell RNA sequencing is the sensitivity to contamination of various origins, of various types. Detergents that might be present in cells that are going to be sequenced by DNA sequencing methods are not necessarily a problem, but they are oftentimes a problem for the mass spec analysis. So that's an important distinction. But then once we have prepared the sample by one of the methods, we need to separate the peptides or the proteins. There can be no deep and quantitatively accurate protein analysis without separation. This has always been the case for all of the methods. And high performance separation is very important. It can be done either by using in liquid phase, by using liquid chromatography or capillary electrophoresis, or excitingly, more recently, there have been a number of solutions to also do this in gas phase that can operate on shorter time scale. And there will be a couple of workshops in this morning session discussing how separation works, making it more accessible for everybody, and sharing some of the recent progress in those areas. The one final thought that I want to leave you about with this section covering the basics, the methods from high altitude perspective, is that there are accessible solutions for all of this. So if you wanted today to start doing single cell sample preparation for mass spec analysis, you can do that using reagents that are widely found in many laboratories. Some of the methods are less accessible, some are more accessible. Similarly with separation, there are commercial, robust solutions which you can start using today. And there are really no boundaries to adopt some of the more accessible methods. And then, of course, the next thing after you have done the sample preparation and you have separated the peptides or proteins is to do the mass spec analysis. And I want to give you a very quick overview of how that works, starting with how a single peptide would look like. Well, a group of peptides with the same sequence. It's not a single molecule, it's a single peptide sequence represented by multiple copies. They look like these features. One dimension here is the separation. We just talked about it, that separation is essential for mass spectrometry proteomics for deep analysis. And then we have multiple isotopic peaks corresponding to the different number of heavy atoms present in the molecules. And now, knowing how a peptide looks like as a peptide-like feature, we can ask, do we see many peptides when we analyze single cells in the forms of ions? And this can be represented as a heat map, where on one side we have the separation dimension, on the other one we have the mass over charge ratios. And in this plot, each color corresponds to a different peptide-like feature. Here they're zoomed in so that you can see the distinctive characteristics, the isotopic envelopes. And the really good news in this context is that we can see tens of thousands of peptide-like features from a single human cell analyzed on the mass spectrometer. And this tells you immediately that the technology has sufficient sensitivity to detect ions from a large number of peptides. And of course, the good thing is that each of these features don't correspond just to a single molecule. They correspond to many dozens, hundreds, oftentimes thousands of copies of that peptide. The challenge then is that simply seeing the peptide-like features is limited because we don't know their sequences. We don't know what they are, and we know that they're peptide-like, but some of them may not even be peptides. And simply by analyzing the features, we generally cannot determine their sequence. Because if you take a peptide and permute the amino acid sequence, you're going to have the same mass, but different sequence. And therefore, just measuring the mass is insufficient. The way to solve this problem is to fragment the peptide, measure the fragments, and then from these fragments, we can determine the sequence. If we measure enough fragments, we can sequence de novo the peptide. Oftentimes, we may not measure enough fragments, and then the statistical analysis becomes challenging. But at this point, if you're following the description, and if I'm describing the situation well enough, you might be asking yourself, how can we possibly analyze all of these tens of thousands of peptide-like features if we're going to be fragmenting peptides one at a time? Because it takes time if we're doing this serially. And this is really one of the key things to understand, that I think to enable you to understand much of the progress happening in the field. What are the various solutions that computational and otherwise that colleagues are coming up with to deal with this challenge, to be able to determine the sequences of all these peptide-like features that you're detecting. And I would say that, at least in my opinion, having the information from the MS2 fragments is very useful, and traditionally has been the most reliable way of determining the sequences with confidence. So this very rich ion map can be sampled in a couple of different ways. One approach to sampling it is simply to use a heuristic of picking the most abundant peptide-like features. And that is convenient, but as you can imagine, limited, because the most abundant, they're not the ones that we're necessarily most interested in. And they may not even be peptides in some cases. So we can waste time on analyzing something that we're not interested in. Another approach is to isolate in large windows many hundreds of peptide-like features, fragment them, and analyze all of them. And because this isolation is independent from the precursors that have been measured, this method is known as data-independent analysis. And you can see how this method can begin to at least address the challenge of how we might be able to analyze MS2 fragments from all of the precursors. Not necessarily interpret, that's a different challenge, we're going to talk about it, but at least we collect the data from all of the detectable fragments. Or alternatively, one can choose a subset of peptide-like features that are most relevant to the biological analysis under interest. So all of the existing mass spectrometry methods can be categorized based on the degree to which they multiplex either samples or peptides. How many peptides are analyzed at the same time, how many single cells or otherwise samples are analyzed in the same time. And if we don't use labels, if we don't label the peptides, then we have the label-free methods that can either analyze one peptide at a time, so each feature is isolated as one at a time, fragmented, and the spectra analyzed. And this has the advantage that the MS2 spectra is simpler. Alternatively, one can use the data-independent acquisition to isolate many peptides simultaneously in a wide window, fragment them and analyze them. And this has the advantage that now you increase the throughput of simultaneously analyzing lots of peptides, but has the obvious challenge of having much more complex MS2 spectra and needing to deal with that inherent complexity in the data. And then one could also use labels to enable the analysis of multiple single cells at the same time. And these labels could be isobaric, as in the case of tandem mass specs or TMT, and combined with analyzing one peptide at a time. Or in the case of non-isobaric labels, recently it has become also possible to simultaneously multiplex single cells and peptides. So with this very brief introduction to mass spectrometry, I want to give you a quick flavor of what results look like from single-cell mass spectrometry analysis today. What is the state of the field? And I don't have enough time to do a comprehensive review. I'll simply highlight some of the important aspects of the analysis. And we are going to talk about the depth of proteome coverage, about throughput. But first, I want to start by giving you a flavor of what the raw data looks like, because it's easy to count number of data points, but if those data points are poor and noisy, what's the meaning of all of it? It's just numbers and statistics. So let's look at the raw data. What do the raw data look like? And actually, we'll look at the raw data. I'll first say that the analysis can be performed both on old and new instruments. And in this case, we have applied exactly the same method using either a very modern, very powerful instrument, timsTOF SCP, developed by Bruker, or using a very old instrument, more than a decade ago old instrument from different mass detector, Orbitrap mass detector. And both old and new instruments can result in high quality data. Clearly, the newer instruments perform better. They give deeper coverage. So if you have the resources to buy the latest and the best instrument, that's what you should do. But if you don't, you can still use the older instrument and get usable data. So let me show you some data from this old archaic instrument of more than a decade ago. So this is data acquired by Jason Derks of single cells where we had each cell being labeled with a different non-isobaric mass tag, and we are looking at the peptide precursors for one peptide and corresponding to the different mass tags. So here you can see clearly the isotopic peaks corresponding to different number of heavy atoms in the molecule of this peptide, and you can clearly resolve those labeled with one mass tag, the other, and the third mass tag. This is a view that I think is even more convincing, at least for me, is we can now extract the ion current that corresponds to those peptides in single cells. So let me orient you in this plot. Here on the top, we show the ion current of the peptide precursor from a single cell. So this is a peptide sequence, and this peptide is eluting from one pancreatic adenocarcinoma cell, and you can sample it at multiple time points, and you see the elution peak. And this is another cell, monocyte cells, in which the same peptide, the elution curve, is being detected. Then you can also fragment this peptide, because if we want to identify it, we have to fragment it. And we can trace the elution curves of the fragments of this peptide. And you can see that their retention time coincides with the retention time of the peptide. And this can give us confidence in the identification. And similarly for other peptides, you can find this peptide is more abundant now in monocytes as compared to the pancreatic adenocarcinoma cells. Now, one thing that I find quite reassuring when we have this data is the ability to derive a degree of an estimate for the reliability of the data, a degree of confidence, because in principle we can derive relative quantification from multiple different scans and from different levels. So we can take this MS1 scan and obtain one estimate for what is the relative abundance of the peptide between those two cells. From another scan we obtain another estimate. From a third scan we obtain another estimate. And now we can take the fragments and do exactly the same analysis and obtain another set of estimates. And if all these estimates agree, as in this case, we can have fairly high confidence in the data. And this is reliability at the level of a single peptide measured in single cells. It's something that I find very gratifying. This is an exemplary case. The data will not always look like that. Sometimes there might be disagreement, and that is really good to know. If our data are noisy, we better know that they're noisy, but if they're not, you can have this confidence. So in terms of quantification, there is certainly the capability to achieve a very high level of quantitative accuracy associated with estimates of what is this accuracy. So this was a deep dive into the data for single peptide in a couple of cells, but we can look more broadly what the data look like across many proteins, now measured in single cells, here on the y-axis, or measured in hundreds of cells. So this is relative quantification. We're looking for protein fold changes between two different cell types for a large number of proteins, and we see that the agreement is quite good between the single cell and the bulk measurements. Okay, so that was a vignette, I hope, gave you some taste of what the raw data look like. Let's see now what are some of our statistics in terms of proteome coverage. This tends to be a question of intense interest. And the first thing to consider is how many total protein molecules can we sample from a single cell. That's a statistic that is very widely used for single cell RNA sequencing. Usually, various methods are able to count between a couple thousand transcripts per single cell to maybe 20, 30,000. In the case of mass spectrometry, depending on the method that one uses and what does, this number is substantially larger. It's goes from about 250,000 to a few million peptide molecules being quantified per single cell. And this corresponds to somewhere between a few hundred to a few thousand proteins. And you can see what is the distribution of copy numbers per protein. So there's, because of the way we operate the instruments, usually there are no proteins that are detected with very low copy number. They're at least order half a dozen. Most of them are at least, the median is at least a few dozen copies being counted per peptide per protein. And this means that the counting noise, while present for single-cell proteomics, it's much, much lower than the counting noise in single-cell RNA sequencing. So it is higher than in bulk proteomics, but it's much lower than in single-cell RNA sequencing. And of course, this number here of number of proteins per cell is a metric that the community has traditionally been very focused on and wants to increase, and that's a good thing. But we have to keep in mind that it's not the sole or the most important metric, because there are also all sorts of questions that we have to ask to put it in context. One is, what is the confidence in the number that is being reported? What are the copy numbers sampled per protein? What are the systematic artifacts and noise associated with those measurements? So in many ways, I think it's not the most important metric to, at least not the metric to emphasize in isolation of all the other ones. But that being said, it's clearly an important one. We clearly want to increase the number of proteins that can be reliably sequenced and identified from single cells. And I have argued in this perspective here that we have the potential to increase the depth by at least an order of magnitude by using the existing technology, just improving our data interpretation and analysis. And this is something that I'll be happy to talk more about if you're interested. Okay, so I talked a bit about the quality of data that we get, about the depth. And the next question is, what is the throughput? How quickly can we generate these data? And this depends substantially on what method is being used. In some cases, it's about 10 cells per day. In other methods, it can go to a few hundred single cells per day. And this is just the throughput of mass spectrometry. But there's a very important aspect. What about the sample prep? The sample prep actually used to be the main limitation because it was hard to do it reproducibly day in, day out. At this point, with the latest generation of sample prep methods, their throughput substantially exceeds the throughput of mass spec analysis. So mass spec analysis has become the bottleneck, not sample prep. And I'm confident that as we increase this throughput here, which is coming, the sample prep methods will easily scale to also increase the throughput. Then it's not only the number of cells that one analyzes, because if we wanted the largest number of cells, we could just do flow cytometry and analyze more cells, but at the expense of measuring only 1 or 2 proteins per cell. It's also the number of data points. And at the moment, we can generate about 500,000 data points per instrument per day. And speaking of the throughput, I should say that one of the most important determinants for what is the throughput of a method is the degree to which it's performing multiplexing, either at the level of the single cells, analyzing multiple single cells simultaneously, or at the level of peptides, analyzing multiple peptides simultaneously. So the more one multiplexes, the larger number of data points we can acquire per unit time. And if one multiplexes both of those things simultaneously, there is a potential for multiplicative increase in the throughput, which we have illustrated here schematically with this pyramid. And we are certainly very excited about climbing this pyramid. At the moment, we have a proof of principle demonstration of this point, but I'm really eager to get more towards the apex of this pyramid and believe that this is very much feasible. Again, that can be another great discussion point if you have questions about it. There are a lot of resources that you can start using in terms of data if you're interested of analyzing data, but you cannot yet generate them in your lab. You can certainly visit websites with deposited both data and metadata. They want to put here the emphasis on the metadata, because if the data are deposited but the metadata are not, it's not very useful beyond just taking space on hard drive somewhere in the cloud. Metadata is very, very important to be deposited. And since the first conference, we have analyzed about 5,000 single cells, which is a low number, but it has taken us a while to get the methods up and running. A lot of the delays have been on the side of sample prep. But just to give you a sense of what's coming forth now, Saad Khan, a PhD student in my group, has analyzed 1,000 single cells just for one project. And this is small. We think that we can easily scale and increase this. So we think a lot of the growth that we had seen in single cell RNA sequencing is going to also come to single cell mass spectrometry. Not necessarily at the same rate, at the same scale, but we are going to see very large data sets coming forward with thousands and tens of thousands of single cells analyzed. And we've also worked very hard with the community, many of you who are present here and virtually, in developing guidelines that can help the broad adoption of the methods that have been developed in a way that encourages good practices, in a way that generates data that can be reused, data that can be evaluated, data that can be easily shared. And we would like to welcome all of you to ask questions and suggest additional guidelines. This is a two-way conversation. It is not just one set of guidelines that is going to stay there forever. It's an initial set of guidelines. And we would like to get more suggestions and more feedback in terms of what additional things and standards should be added to facilitate the healthy growth of the field. I'll stop here, and I hope you can help me express the questions encoded in the DNA, because we're interested here at the proteome scale.
Wonderful. Any questions for Dr. Slavov? We'll take questions from the live audience first, but we have a few in the Zoom chat.
I see a question here, Erwin.
Hi, beautiful summary. Thanks a lot. Also very nice results with the plexDIA. I'm going to talk to you a lot the next 2 days, but maybe just for this purpose, I was very interested in your MS1 versus MS2 peak comparison of your top three. So were there any conclusions in terms of overall discrepancies between the two? I mean, there's a lot of discussion in the field of should you use MS1 quant with DIA or MS2 quant, and I guess you have a perfect data set to evaluate that, especially with a single cell resolution.
Yeah, so this also depends on the method, how that is being designed, because we can have a data acquisition method with DIA, where we spend most of the time in the MS1 level on MS1 scans, or we can spend in another kind of method most of the time on MS2 scans. So in the first case, the MS1 data will be better. In the second case, the MS2 data are going to be more quantitative, just because of how much we acquired, what is the temporal resolution. Specifically in the context of single cell analysis, we think that MS1 level is likely to give us much higher sensitivity, because peptide ions have high charge. High charge means better detection. They're not yet fragmented. Fragmentation is always a lossy process. Abundance of fragments is always going to be lower than of the peptide. So we think that MS1 level quantitation is very exciting and preferable for single cells. But that being said, I don't see it as being lopsided, just MS1, we should use MS1, ignore MS2. I really want to have both. And MS2 can be extremely useful. For one thing, at the MS1 level, one could have interference that severely distorts quantitation. And the way to know that is if we also measured MS2, because the interference at the MS2 level is very, very unlikely to be the same. So if both provide the same information, I can have very high confidence in a data point in a single cell for a single peptide. And to me, that's very gratifying. So we think both, but in many cases, I think the MS1 level is going to have a clear advantage for single cell analysis.
Thank you.
Thank you for this presentation. I think it's a really good introduction to single-cell proteomics. I think I will start with a comment, and one thing we might forget about single-cell is the dynamic range of the proteome. If we talk about HeLa or if we talk about other cells, the dynamic range can be completely different, and I think that is something we are forgetting for now, because it's really a complex parameter to take in account. And my question will be, so we talk about acquiring more data, more cells, but the next bottleneck, if not MS, it will be data. how to deal with data set containing millions and millions of cells, how to normalize, how to correct batch. Do you think about that? What are your perspectives for that?
We have thought a lot about it, and the gentleman sitting behind you has also thought a lot about it. It's a great question. Data analysis is very important. I did not cover it. At the moment, there are a number of, and the gentleman in front of you has thought about that too. At the moment, there are a number of pipelines that you can use, open source pipelines implemented in either R or Python. And that's a good start. At the moment, I think that's adequate. I particularly like the pipelines that allow you to change various choices of analysis steps and see what the result of that is. I think that's very powerful and very useful. Simple, but useful. Also good things. As it scales further, we certainly will need to scale those pipelines. I don't see that as being a major challenge. I think it's an important problem to engineer and solve well, but we can also piggyback and take advantage of many of the solutions for single-cell RNA sequencing that have already experienced this challenge of massive large-scale data sets. And much of that infrastructure, many of those solutions can also be adopted to work with proteins. So I think there is important work to be done there, especially in terms of benchmarking. One of the really important urgent things in my mind on that side is characterizing what are the noise properties in single-cell protein measurements. We know that there was a lot of work, hundreds of papers published on that for RNA sequencing in the early days. We don't yet have a single focused dedicated study on that question. So that's a useful thing to do. And then incorporate this into pipeline so that we can do it more as a rational engineering. We know what are the noise properties. So there's certainly space for improvement, but I would say at the moment, there are adequate solutions that people who want to start doing analysis today can start using and can start getting usable data.
Thank you.
Any other questions from the room?
Thanks, Nikolai, for a fantastic overview. Just with regard to the plexDIA, I wonder if you've done, I know you've got a strong mathematics background. I wonder if you've done the theoretical calculation of how far you could push the non-isobaric labeling.
Yes, we have thought very much about that because we are very seriously intending to climb that pyramid. And probably the most useful empirical data point for how far we can go comes from our analysis with the bulk samples, where we were able to detect and quantify over 200,000 peptides. And if we go back to single cells, where per single cell we quantify maybe 10, 20,000, that means we can increase the plex by at least 10 times and not hit the limit. So we did not see, we certainly had some level of interference. There's always level of interference in DIA label-free plexDIA, but we did not see a very steep increase in the level of interference at that point, at least when implemented on orbit traps at the MS1 level. And because of that, I'm encouraged. I know we haven't yet reached the limit. Where that limit exactly is, I don't yet know. We have also tried to simulate internally, even it's completely simulating the orbit trap transients, resolution, and all of this. So there will be a limit, especially for the bulk samples. My guess would be around 10 plex for bulk samples. For single cells, I think it's much higher. It could be 100 or higher plex, certainly what we hope to achieve. And at that point, the throughput of single-cell proteomics will in many ways catch up or exceed throughput of single-cell RNA-seq if we get there. And I hope we will.
Any other questions? So we have a few questions from the Zoom chat. Fatma asks, what is the advantage of studying single cells, just in general?
Well, if you study complex human tissues, they're made up of cells that are functionally different. And we want to be able to analyze those differences. If you grind them up and study the average abundance of proteins in lymphocytes and tumor cells, you cannot understand whether the proteins that you measure came from a lymphocyte or from a cancer cell. And that's important for the biology that you're investigating.
Stella asks from the Zoom, are those samples referring to the plexDIA figures from in vitro cultures or extracted from cancer tissue? In the latter case, how well do you think the single cell separation and identification can be?
So the data, the raw data that they showed came from in vitro cultured cells, but we have analyzed primary cells from human tissues. In fact, Andrew might show some of that data. And the data don't look very different. The main challenge with analyzing primary cells in our hands has been with sample prep. And that was dramatically solved by the use of the droplet sample preparation that Andrew is going to talk about. So at the moment, the main challenge of studying primary cells compared to in vitro culture is that some of these primary cells are quite small, such as T cells and B cells. They're smaller than the typical cell from a cancer cell line, and therefore the coverage is going to be lower. There would be fewer protein copies measured. But other than that, there's nothing that's fundamentally different.
Nicole asks, what are the most important scientific questions do you think that single cell proteomics can solve?
So this is a very general, big picture question, and I'll give a very general answer. I think starting from 1970s or so, we have been developing methods to be able to make more and more detailed mechanistic measurements of cells grown in culture. And about 10 years ago, we shifted from cells grown in culture to studying cells from complex tissues, such as from patients, in large part because of RNA sequencing methods. And these methods have really allowed us to descriptively classify all these primary cells into lots of categories, into clusters. But our tool set to do anything more than that is incredibly limited. And if we want to have some of the tools that we have for self-studied in vitro, for example, if we wanted to measure enzyme activities or protein shapes and protein interactions, if we wanted to have direct evidence for ligand receptor interactions, the only way to do that is to use proteomics of one type or another. So I think the big promise for me of single-cell proteomics is turning single-cell biology into a more mechanistic quantitative discipline in a way that biology was with in vitro samples, and in a way that biology has now sort of diverged as we have focused mostly on measuring abundances of RNA, DNA molecules in clustering cells. It's a long-winded answer, but to such a general question, I couldn't resist giving a more general answer.
We have another question from the room.
I think you already mentioned some of the importance of single-cell proteomics. Can you be a little bit more specific about how the single-cell proteomics can kind of complement the single-cell RNA-seq when you try to understand the mechanistic, suppose cell proliferation, those kind of things, how single-cell proteomics can complement single-cell RNA-seq?
Yeah. So I think the one very approachable aspect that we're excited about is inferring post-transcriptional regulation. And Megan, sitting just in front of you, is a good person to talk with about that. So we think that by comparing the abundance of proteins and RNAs, as long as we can account how much of the divergence is due to noise and technical factors, we can attribute the remaining level of the divergence to post-transcriptional regulation, perhaps even to mechanisms. So we think that's really the most obvious and straightforward way of combining them. You could also extract time information in the same way that with RNA velocity, the spliced and the unspliced form provides information for the vector field where the cell is going. In a similar way, the RNA changes are going to precede the protein changes, and that can give a temporal component to the measurements. These are two specific possibilities, and there are certainly many other ones.
Claudia asks, you mentioned that single-cell sample preparation is not a bottleneck anymore, but do you think we have the optimal sample preparation available for all the diverse biological applications out there? Don't you think that, especially due to the new application strategies also, the sample preparation has to evolve continuously? For instance, and I'm commenting here for like tissues.
Yeah. So not being bottleneck for many of the applications certainly doesn't imply optimality. It certainly will continue to evolve. I don't think we have the final ultimate solution, but my comment is more from the perspective of having lived through years. Ryan can account to that as well, of years when we did have the ability to do the mass-spec analysis, but what really limited the number of cells we could analyze, what we could do was just doing the sample prep. So from being a primary bottleneck, it has moved to not being a bottleneck. Doesn't mean perfect. Nothing is perfect. Everything that we talk about here can be improved massively. One aspect that has been relatively little developed and discussed in the context of single-cell proteomics is the spatial component. We certainly haven't done that. There is some interesting work with laser capture microdissection, and a lot of that is not done at single-cell resolution. Some of that is close to single-cell resolution and can be improved further, but I think that there will be a lot of development on the front of adopting sample prep for spatial applications. As we get to 100 plexDIA, as Andrew asked, we are going to need to further increase the throughput of sample prep. At the moment, it's not limiting, but as we advance throughput of mass spec, sample prep is going to become limiting again. So, and then as we define optimal, we really have to define our metrics, what we mean by optimality. And Andrew is going to talk a bit about that. He has some really good slides of specific, concrete, measurable quantities that we can use to evaluate how good or how poor sample prep is.
So you just answered another question asked in the chat. And if there are no more questions from the room, let's thank our speaker and welcome our next one.